This conference will now be recorded.
Okay, hello everyone. It is 1 o'clock on the East Coast and we are going to go ahead and get started with our presentation. My name is Jessica Enix and I'm with the ACIL staff and I would like to welcome you to today's presentation. Before we begin, I'd like to review a few points of protocol so that you'll get the most out of your webinar experience. Remember, you are listening to the webinar via a conference service. If you are disconnected, repeat the steps that were given to you in the registration email. In order to reduce background noise, please be aware that all participant lines are muted throughout the presentation. If you have any questions during the presentation, please type them in the chat box on the right-hand side of your screen. The presenter will address questions at the conclusion of the presentation. Please note that today's program is being recorded and will be available on the ACIL website at the end of the day today. Now it is my pleasure to introduce our presenter, Mr. Doug Stevens. Doug Stevens is a principal scientist in the scientific operations team focusing on food and environmental applications and markets. In his 30 years with Waters, he has consistently worked on the analysis of small molecule analytes using GC and LC with tandem, quadrupole, and TOF mass spec for both routine quantitative work and qualitative work, such as unknown identification. His most recent work has been in the analysis of cannabis and environmental samples. Ladies and gentlemen, please welcome Doug Stevens.
Thank you, Jessica. And of course, you can all see the title, but before I just dive into the presentation, I'll give you a little background about what you're going to see that hopefully will help explain why the adoption of GC-MS as opposed to GC-MS has been steadily increasing in food and environmental applications. So we'll start the presentation. There'll be a few slides on the background about food and environmental applications, including some of the regulations that have evolved across about the past 13 years since the commercial introduction of this type of technique. Then I'll go into the technique itself and compare and contrast the technique to EI. So the technique we'll be talking about is atmospheric pressure GC. So APGC is the acronym you'll hear, and that's the Waters product. You'll also see the term GC-APCI, which is the generic term for the technique. And we'll compare and contrast some data from EI, which is the more traditional, older style ionization for GCMS, to atmospheric pressure chemical ionization. After the technique coverage, we'll go on into an applied example looking at pesticides in cannabis extracts, and that will cover a little bit on the sample prep as well as some complementary LC-MS analysis. Then on into semi-volatile compounds in environmental matrices, and right before we finish up, I'll go into some alternative carrier gas using nitrogen instead of helium. All right, so here we go. So first, looking at the types of analytes we'll be focused on in this talk, they're all in the category of semi-volatile organic compounds. And as a step back to look at all the GC amenable range of compounds, we'd also find gases or very volatile organic compounds, and things like solvents in the volatile organic compound category. In this case, for semi-volatile, we'll be focused on things that have boiling points generally in the range of 100 to 600 degrees C. And to give you an idea of something that might have a boiling point around 600 degrees Celsius, the system has been used to analyze compounds up to a carbon chain length of C80. So that's kind of the upper range. So when we talk about semi-volatile, these are not compounds that in layman's terms would be described as volatile at all. But when we introduce them into the hot injection port of a GC and force them through the capillary column with some helium or other carrier gas, then they become volatile enough and amenable to GCMS. One of the goals for this type of work is often in environmental work is broad characterization of a site. So if, for instance, we know a site is contaminated with dioxins or PCBs, we can do a narrow focused analysis for those compounds. Commonly, though, we encounter industrial sites and environmental sites that need broader characterization of any inputs of synthetic chemicals that we might want to look for in the semi-volatile class. So that can span broad range of compound classes and analytes. And it's a common high volume analysis in a lot of environmental labs. Going on to where kind of food and environmental overlap, you can see this example. There's a nice citation at the bottom of a fairly recent article that does an excellent job covering why across the range of both food and environmental, we find ourselves looking at a common set of analytes in different matrices. And in this case, what you see is if we follow a pesticide from being manufactured, we'd find it in a quality control environment and testing in the manufacture process. We'd find it being studied as an agricultural residue after application, often to a food crop, and be looking at, as an environmental analysis, the effects on plants and animals, as well as small organisms in that environment, along with transformation products of the pesticides. So the analyte list grows and grows. If we look at it from the food context, we have additional pesticides that might be applied to storage areas during shipment and storage of food materials. The food contact materials themselves, inks, dyes, and plastics that might be in touch with the food product. And all of these can lead to a need for broad characterization of analytes in a huge range of matrices as well. The connecting factor across all of these is often also the effects or negative effects on human health of these various compounds as they go through the different compartments in the environment or through different food storage and production issues. Now, I mentioned earlier that it was about 13 years since the introduction of APJC. That was at PitCon in 2009. If you look at this 2014 regulation from the European Union, by this point, 5 years into that system being available, it had already been included in a working group in Europe where the proposal was made to do confirmatory dioxin analysis in food and feed using GC-MS as an alternative to the traditional EI high-resolution magnetic sector work. And part of the reasoning behind that were the common reasons you'd find for the popularity of tandem quadrupole in target analysis. And that's the ease of use and sensitivity, as well as all quality control criteria being able to be matched. So a working group formed in Europe. They approached the regulators and said, we'd like to replace the magnetic sector and allow the use of EI or atmospheric pressure GC-MS as a confirmatory analysis for dioxins in food and feed. They went through multi-lab validation study using multiple matrices, and in the end, that resulted to the change in this legislation in 2014, as well as a subsequent update in 2017. So again, I'll point out the main change here was to go from magnetic sector, which tends to be large floor standing instrument that requires high expertise in the operator, to benchtop systems on EI and atmospheric pressure GC-MS that are easier, smaller, and less expensive to operate. The United States was a little bit behind Europe in this respect, but in the environmental field, we have the Clean Water Act that the EPA has as part of their charter. And this allows for alternative test methods. And in 2020, so now 3 years after that previous updated European method, SGS Access, working in a collaboration with EPA, came up with an alternate test method for dioxins and furans in environmental matrices. And that's what you'll sometimes see abbreviated as ATM 16130. And this again allows for confirmatory dioxins analysis to be performed with either EI or atmospheric pressure ionization, but GC-MS as an alternative to high resolution magnetic sector MS. And again, with the drive behind this being to reduce the cost, to reduce the size, and maintain support as more and more magnetic sectors come to the end of their manufacturing lifetime. So here what I've shown is the variety of the sources. So across the top line here, what you're seeing are different sources that can go on the different types of mass specs. Here we'll focus on the tandem quadrupole, but there are single quadrupole, time of flight instruments, and ion mobility systems that all these techniques would go across with a corresponding range of different chromatographs. The GC, we have LC and micro LC, as well as SFC or supercritical fluid chromatographs that could go on the front. What we're going to be looking at is this pair now highlighted in green. The top portion says APGC, that's our ionization source, and the lower section shows the mass spec on the left-hand side, that's the tandem quadrupole, and the GC on the right-hand side. And we'll have another look at this a little bit closer up as we go along. So if we open that door, that source door swings open. It's atmospheric pressure, so no venting of the system is necessary. This is what it would look like inside. So on the right-hand side, I'll draw your attention to the diagram of the source. In from the right-hand side, we see the GC column coming in from the right into that region where there's a reduced volume insert that goes into this source. The source itself has a large open architecture, which makes it nicely amenable for electrospray or APCI with LC. And when we go to GC, we add this insert listed as the ionization chamber. Inside that chamber is a corona pin. Again, the generic technique is GC-APCI. And the makeup gas indicated there is nitrogen, typically at a flow rate of around 300 mils per minute. So that nitrogen and the analyte entrained in that nitrogen and helium flow towards the corona pin. I did in large print the cone gas, which flows in the opposite direction to balance the flow around that area of the corona pin, as well as to control ionization. In the upper left, what you see is kind of what you would see if you were an analyte flying out the end of the GC column. And you may be able to just make out the corona pin tip right in front of the first orifice into the pumped region. Below the word cone gas, you'll see a little green circle, and that's indicating where the cone gas flows up and flows counter to the makeup gas. Note that this whole region outside the blue ionization chamber is an atmosphere. So doing this switchover doesn't require venting the system, whether you're going in to go do standard maintenance or to just add water into the source, as is shown in the right-hand side. So the right-hand side shows the way we introduce water as a reagent when we want to encourage protonation. We'll talk about ionization next. And that, what you're seeing there is 3 2-ml vials, standard just autosampler vials with capillaries through the top that allow a constant amount of water out for on the order of 1 to 2 weeks if we fill those each up with 2 mils. Now on to ionization type. If we're looking at ionization type and controlling it with cone gas, these are our two considerations. We have charge transfer and protonation. And I'll start with the bottom actually, because when I say GC-APCI, I feel that some people attending might already have a pretty good understanding of LC-APCI. So protonation is common between the two techniques. And if you look all the way to the right in red, you see M plus H plus. So what you get when water is added into the source is the protonated form of the molecular ion, provided the analyte has a proton affinity higher than that 691 kilojoules per mole listed down in the left-hand corner. Water is the reagent, and this is a thing I specifically point out because it's quite different than a vacuum source classical GC system that people are used to. In vacuum source GCMS systems, you may spend huge amount of times trying to get the water out of a system to get it to full dryness, as water can be an indication of leaks. In a vacuum source system, that's a real problem. Here, we're intentionally adding water as a protonating species because the system is otherwise quite dry, right? We have 300 mils per minute of dry nitrogen flowing in, plus 1 to 2 mil a minute of helium. Without this provided supply of water, we would get charge transfer. And there are occasions where we want one or the other or both types of ionization. So for instance, now drawing your attention to the top of the slide, where you know it says IE equals 15.6. So for nitrogen as the reagent, when we do operate in dry source mode, that creates a situation with about 15.6 EV ionization potential. The majority of compounds that I've already discussed or that we'll cover will have an ionization energy of 12 or less. So anything under 15.6 will go ahead and ionize. And that's the vast majority of analytes that will also volatilize and come through the GC column. Now, a distinction here, though, is without a source of protons, like the water becomes, what we get is a different form of the molecular ion. And you can see it indicated as M plus dot. So rather than becoming positively charged by adding on a proton, it becomes positively charged by giving up an electron to the plasma. So in that form, it's a radical cation, just like the molecular ion in EI systems when they're present. But this is at a much lower energy process. So it's a high efficiency, low energy process, but it's also well established. That's why I have the references across the bottom, across the kind of over 50 years of chemical ionization references. You'd find charge transfer. It's also referred to as charge exchange, all the way back into the 60s and fundamental studies by Munson and Field. So there's nothing been invented in terms of the ionization type here, but it wasn't commonly used in vacuum source systems. And again, remember there's 300 mils per minute of nitrogen as the reagent species here, which isn't really compatible with a vacuum source system. So that's one of the contributing factors to charge transfer being maybe a little less familiar for GCMS people. But one thing we can see when we do vary the cone gas, if we look at the reagent ions, which would be common if we were doing methane CI in a vacuum source, what we can see is even just looking at the reagent ions, we can see as we go from low to high cone gas, the CG, we see different reagent ions. And that's a reflection of what's going on in terms of the gas phase chemistry in the source. It's probably a bit more meaningful to actually look at that on real data. So now let's take one of the pesticides. This data just generated last week. And we have a charge transfer spectrum of dieldrin versus the EI NIST spectrum. And the stark contrast is that you see in EI, most of the ion current is in that 79. So that's a low mass peak of low specificity. So what we're doing here is we're creating, in this picture, it's pretty obvious there's a high relative abundance of the molecular ion. And what you'll also notice is that the 380, which is the base peak in the molecular ion cluster, isotope cluster, is exactly the same, right? Because it's the same form of the ion, The 378 is actually the molecular ion, the monoisotopic molecular ion, and that's the same in both spectra, but the relative abundance is starkly different. However, since we're on a tandem quadrupole, we can go ahead and fragment that to a user-definable extent. And if we do that, you can see the dry source or charge transfer spectrum, APGC spectrum, shares all the same ions in common with that NIST spectrum. So we're able to go ahead and get good correspondence to historical EI data through the use of the dry source mode. If we now compare and contrast the dry versus wet source mode, what you can see for that same compound, DFTPP, which is a common tuning compound for semi-volatile analysis, we can see that there are distinctly different ions for the different ionized forms, since the ionized form dictates how the compound will fragment when we do CID in the collision cell of the tandem quad. If you look closely, you might also notice that there are some ions in common. The 69 and 77 are present in both spectra. And this is a nice kind of subtle indication of why, when we do MS-MS, we prefer not to use MRM transitions to very low masses because they can be less specific. So if we go on now to looking at a specific example of the use of this technique, this is a good match for explaining why are people bothering more and more with tandem quadrupole in both LC and GC-MS studies on environmental and food matrix samples. There's a tendency to want to do broader characterization, look at a wide range of chemical compounds. As we know, pesticides can be neutral, acidic, basic, and span a broad range of chemical class. Pesticide, of course, is its intended use as opposed to its chemistry. So to match with that, we often have a very generic sample prep. And in this case, this example for the cannabis extraction is very simple. It's basically ground up and shaken with acetonitrile. We're going to take a portion of that for both LC and GC cleanup after this initial extraction. And there was an application note published on this, and I've included a link. This initial step amounts to a 20-to-1 dilution of the sample from 0.5 gram into 10 mils. And if you look on the right-hand side, what you can see is that in GC-MS, if we run the raw extract in full scan mode, we can pick out all the cannabinoids and THC that are extracted during this simple process. If we then have the MRM data from the target analysis of the pesticides shown below that, you can see that those elute in the same retention time range. So there's overlap of these high abundance cannabinoids in this very raw extract. So after extraction, we go on to an additional cleanup. This is a dispersive SPE cleanup. So again, fairly simple, where we take 100 microliters from that previous extraction and add it to the tube with the dispersive SPE cleanup material, centrifuge, and then transfer the organic layer to the vial. This now will amount to a total of about a 200-to-1 dilution, and the purpose for it is listed here. It's going to remove the fats and pigments, as well as up to 75% of those cannabinoids shown in the previous slide, while letting the relatively neutral analytes pass through. It's also going to remove the acidic compounds, and this all adds up to making a nice aliquot for compatibility with GC-MS. But it amounts to a 200-to-1 dilution. Now, furthermore, for certain compounds like the cyfluthrins and the cypermethrins in the pesticide category, when we get something that might be a 10 nanogram per mil standard, often that's the sum of these 4 peaks for the cyfluthrins, let's say. So each one of these is 1/4 of that 10 nanograms, and that means for each individual peak, we're looking at what amounts to an 800 to 1 dilution. So to get the broad coverage with a generic cleanup, we end up with a series of stages of dilution, including among the isomeric species that we have among the pesticides. So sensitivity is very important as we then look into specific analytes extracted from cannabis in this study. These are just a couple chosen as examples, the azoxystrobin and fensulfathione from 1 to 500 parts per billion. So now if we look back to the previous slide and we think divide by 4, for the limit of detection for the cyfluthrins, we're down into the 250 part per trillion range. So sensitivity is very important here as we apply these generic sample prep methods to a broad range of compounds. So because of that, it's important to evaluate recovery. Recovery here was evaluated from 20 to 50 and 100 ppm levels. And shown here is the range of over 30 compounds being within 70 to 120%. So overall, in the complete method here, we were looking at 105 compounds. There was some overlap between LC and GC. And Replicate showed that recovery of 100 out of 105 in this AOAC guidance method were recovered in the 70 to 120% range. There were 4 that had lower recovery. Those were in the LC portion of the analysis. They were just a bit low, 60 to 70%, and still met the limit of quantitation for action limits that were required. There were a couple of compounds that degraded quickly. However, the thiophonate methyl was still able to be measured with approximately 100% recovery in the form of carbendazim. So a simple generic extraction allowed broad compound coverage, showed robustness for pesticides, and mycotoxins were included in the LC portion, and it met or exceeded the AOAC requirements for 100 of the 105 compounds. I should point out too that both of those analyses were performed on a single mass spec tandem quadrupole instrument. This is what that instrument looks like, and this is kind of the stopping point where we'll go on into semivolatiles analysis. I wanted to point out again in the configuration where these various components, the source itself, we saw the inside in the corona pin, indicated behind that black door in the atmospheric region. In this next study of semi-volatile compounds, we're again running in a dual chemical ionization mode where we have both charge exchange and protonation occurring. We have a 100 to 1 split injection and a standard 5% phenyl column, but I'm not going to read through each set of conditions for you. I just want to show you that these are some common things. It's a 100 to 1 split injection and an intentional dual CI mode. As we went into method development here, what we did look at is whether negative ion APGC would provide an avenue to get compound coverage, because we know there are some chlorine substituted aromatic compounds in particular. So that was evaluated, and very quickly you can see that it's about 3 orders of magnitude less sensitive in negative ion for the compounds in this 8270, which is an EPA method number for semi-volatiles. And this is a ResTech standard mix of about 76 compounds. You can see in that area from 4 to 6 minutes in particular, the negative ion data shows virtually no response for some of the compounds. So pos-neg switching was not an option or an alternative to the dual chemical ionization mode. Even with the high specificity of MS-MS, there are requirements for chromatographic separation of minimum criteria for these isomeric species. So you see these 2 PAHs, and the method specifies a minimum of 50% valley. So at the very beginning, of course, with a brand new column and standards, we were shooting for a 20 to 25% valley so that as we trim the column, we'd be able to maintain that 50% minimum. And this is at 2 mils a minute with a 100-to-1 split. Again, this was published in April of this year as an application brief, and so I've included the link here for more details. As part of the method development, this work included the creation of a database. It's called a Quantitative database. And you can see here it includes CAS number and name of compounds, because some of these compounds do go by more than one name. And so the CAS number can be really helpful in tracking down specifically which compound you're looking at. And then the ion information shown on the right here in the Quantitative database includes multiple transitions. Commonly, there'll be 2 to 3 transitions in the database, but there is room for more, and it's possible to modify this, create a user-based version of these, and add additional transitions if that's required or desired by the user. But what this does initially is it makes the startup and implementation of a semivolatiles method a lot faster. So looking at this, and I've highlighted this in purple again to indicate that it's charge transfer. And this would be kind of a poster child type of compound for negative ion CI in a traditional GC-MS system. With a couple of electrophilic substitutions on an aromatic ring, you'd expect electron capture to happen very readily. Here what we see is positive ion charge transfer or dry source operation. And you can see the sensitivity. The chromatogram shown here is for 1 picogram on column. The curve shown on the right shows the quantitation from 50 to 550 femtogram to 500 picogram. So 4 orders of magnitude range with good linearity, and that's replicate injections across a little over 30 hours of time from the same vial. So here we have initial indication of very good quantitative performance. If we look across some of the different types of examples, another compound with an aniline, so now we start to add in a nitrogen containing compound. Again, we have that core of about 100 femtogram to 100 picogram with excellent linearity, indicating that so far for the dry source analytes across this time period, the performance is very good for quantitation. And I included the hexachlorocyclopentadiene because it's a precursor for a lot of traditional organochlorine pesticides. Now, initially, I thought, well, I won't include that because it's not really relevant anymore. But on further research, this compound is still synthesized in metric tons in both China and the United States. So something's being done with it. On an industrial level, releases may happen, and analyzing specifically for this compound is still very potentially relevant. Hopefully not, but potentially relevant for environmental analysis. So now we look at some of the protonation compounds, and azobenzene was the highest responding protonator in the list of compounds for this analysis. And we see very similar performance across the 50 to 100 picogram range of mass on column. So concentration in vial would be 100 times higher. And again, on the left, you can see the response for a 1 picogram mass on column loading of this particular analyte along with its confirmatory ion. For benzoyl phthalate, as opposed to some of the other compounds, we now have something that would be in the class of a food contact material. It's a semivolatile compound. It likes to protonate, so it's got very good performance across over 3 orders of magnitude range. And you can see the kind of signal for a 1 picogram on column loading for this compound, which is the phthalates are frequently of interest for food contact material analysis. The one final protonating species, the benzoyl-A-pyrene, again showing very good performance across the range and having relevance because it's one of the most toxic PAHs. In this case, we have a compound that can have both natural and anthropogenic sources. So it's also potentially in both environmental samples and food during food preparation, certain food preparation types, the creation of pyrines into the sample, into, I shouldn't call it a sample when you're eating your barbecue, into the food product is possible. So now, those are some specific examples called out. I'll point out that across the full range of compounds, there were 76 analytes and 6 internal standards. 85% of them achieved better than 0.99 R squared linearity from 100 femtograms to 100 picograms. So that's significantly more sensitive than tandem quad analysis, or sorry, that's significantly more sensitive using the tandem quad than single quad analysis. And I'll point out that actually 45% of the total had LODs in the range of 1 to 10 femtogram on column. So this is the core of those compounds, with a number of them having up to 10 times lower detection limit. Now, one of the things we wanted to do, because we're intentionally having two forms of ionization occurring in the same space at the same time, we wanted to have a way to monitor the bulk gas stability and the chemical composition stability across time. One of the existing internal standards used in this analysis, the semivolatiles analysis, included a perfect example for this. So we have two transitions for the perylene D12, and they represent the different ionized forms. So we have the charge exchange form and the protonated form both present. And by monitoring the ratio of those across time, we can go ahead and get an assessment of the stability of the split between that ionization. And this about 0.5% RSD, again, over 30, it was 30 to 36 hours of analysis, showed that the chemical composition of that gas flow and the breakdown between the division of ionization between those two forms stayed relatively even and constant. If you can make out that basically the ratio of ionization was fairly even, and that is controllable to an extent by the cone gas setting. So now, part of this work was being done in the first quarter of this year, and we did actually get a notification from our gas supplier that they were exercising the force majeure clause within our contract and might not be able to deliver any more helium for the remainder of the year. There was some subsequent easing on that, but that brought us to a full stop with a quick evaluation of nitrogen as the carrier gas. And so using that same column and just switching to nitrogen carrier gas, evaluating this isomeric pair, we still see sufficient separation between these two PAHs, which are isomeric. When you go in close and look, you do see the peak width did increase by 20 to 25%. So by simply plugging in the same column and using the exact same setup entirely, we do lose a bit of resolution with the nitrogen carrier gas. Previous work done with dioxins now instead of the semivolatiles did actually demonstrate that we can match the resolving power and the separating power and even the runtime very equivalently, along with sensitivity, by scaling the column. So in this case, we have helium versus nitrogen on the exact same sample aliquot, showing some of these isomeric separations within the dioxin class of compounds. But what was changed here was the column was scaled down from a 0.25, 30 meter column to a 0.15, 20 meter column. So the phase ratio was kept constant, but everything was scaled down. Now, when we're talking about these split injections and more dilute samples with a higher sensitivity technique, loading of the column on these more narrow bore columns is not a concern. So we can match the performance in separation with nitrogen as a carrier gas. And we also have the capability to maintain sensitivity. This is, again, back into the pesticides with the semivolatiles. And what this is showing is 5 picograms on column with greater than 1,000 to 1 signal to noise for hexachlorobenzene. I thought that was a good one everybody would be familiar with and give a common translation point. So if we're at over 1,000 to 1 for 5 picograms on column, we're still maintaining a detection limit of 5 femtogram on column or lower with a nitrogen carrier gas for hexachlorobenzene. So across the range of now the semi-volatile compounds, we found that we were able to get good sensitivity, linearity, and reproducibility with this dual chemical ionization mode. The reason for going to lower limits is really not because regulated limits are lowering, but it's because it reduces the amount of sample size from a 1 liter water sample needing to be shaken out to somewhere in the 10 to 100 milliliters. It also reduces the prep time and allows more robust performance of the GC through the use of a split injection. So finally, I'd say APGC already has nitrogen supplied to the system. The nitrogen shown as the carrier gas was actually just split off from the nitrogen that goes into the source, and with appropriate filters added. And that doesn't have to compromise separations or sensitivity. Now, I've got a long list of internal people who are acknowledgements here because this is representative of the work of at least 3 different projects across the last 2 years. And all these people contributed either some data, some background, or some experimental design to the range of data that you've seen included here. So thank you for your attention. And I think now we have the opportunity for question and answer if there are any questions.
Again, if you have any questions, just type them in the chat box. There is a chat icon located at the top right-hand side of your screen. While we are waiting, Doug, thank you for your presentation. That was excellent. And again, as a reminder to everyone, this presentation will be available on the ACIL website at the end of the day today.
Thank you, Jessica.
Thanks, Doug, for that presentation. I don't see any questions in our chat yet, but I have one that I wanted to pose to you. As you've worked with some of our customers that have started implementing some of these applications and using the higher sensitivity of the tandem mass spectrometers, are there any particular challenges that you've run into that you could share with the group here?
Yeah, thanks, Jen, because I know we like to show the very nice sensitivity and linearity data, and the real fact of the matter is, though, as people go to these much lower parts per billion and parts per trillion types analysis, you can run into specific problems because the solvent and the equipment that you thought was clean turns out to have background of specific analytes. And compounds like the PAHs and phthalates, as you might imagine, are some of the common culprits. One of the earlier adopters for the semi-volatiles analysis in particular did find as they went to lower levels, they couldn't get a blank for the compound was phenanthrene, one of the PAHs. Troubleshot, and it turned out that actually the supply of O-rings for the injection port liner were contaminated with phenanthrene. So the fix ended up being having to get an alternate supply from a different vendor. And so we like to look at the rosy and the hopeful side of having lower detection limits, but it does come with some challenges. And labs should be prepared to have alternate suppliers of consumables, as well as the potential for, again, specific background compounds within things like solvents. So not just GC consumables, but alternative suppliers or alternative types of quality level for solvents and procedures for dilution preparation and glassware cleaning. So all those types of things can be part of the implementation of a successful method that now brings you from the parts per billion into the parts per trillion type detection range.
Okay, thanks for that. So I do have a question. Do we need to re-optimize MRM transitions when switching over from helium to nitrogen as the GC carrier gas?
No. So in dry source mode, of course, the nitrogen would is does not become incorporated in the analyte. So in dry source mode, the nitrogen just does charge transfer. So only the loss of an electron happens. So there's no mass transfer. And if you're using wet source mode, the water in the source is what contributes to the mass shift. So that would stay constant. If you switch to nitrogen carrier and you want to do protonation or mixed mode with protonation and charge transfer, then the water still needs to stay in the source.
Okay, great. Thanks for that. Can you comment at all on what are some of the typical maintenance items um, that would need to be done on a system like this?
Well, so the front end maintenance remains the same for the liner and the septa and the injection port components. The trimming of the head of the column may be reduced if you do go from a single quadrupole method where you're doing split less injection to a 10 or 100 to 1 split method using tandem quadrupole. So trimming of the head of the column would likely be reduced because you're reducing the loading of the involatile matrix junk onto the head of the column with every injection. What we do find is that the APGC source itself doesn't require cleaning for weeks to months at a time. And I attribute a lot of that to that sweep of 300 mils per minute of nitrogen through the system as part of the reason for that. Also, the architecture of the source is quite large outside the ionization region where the APGC volume was. And so it's very efficient at sweeping out the less volatile crud. And what I didn't show is there's a relatively large exit place to a pumped region on that atmospheric portion of the source.
Thank you for that. I guess my last question for you would be, in your experience, you've worked with different matrices, both food, environmental, cannabis related. What are your thoughts and insights on how the tandem quadrupole systems deal with matrix interferences and sample matrix in general?
Yeah, part of what I've seen, I should say, I started as a GC single quad applications chemist about 25, 30 years ago. And what I see is largely different is the specificity level in going to MS-MS. So regardless of the ionization being EI or APCI, I haven't seen much difference there in terms of performance in things like specificity. Sensitivity comes with the high abundance molecular ion for the atmospheric chemical ionization, but they both appear to have similar dynamic range and robustness. So it's the sensitivity there, and then the specificity of MRM. And if you look historically at where MRM really took off and tandem quadrupoles, it was with LC analysis, where there's just a mess of things being sprayed into an electrospray system. So by going to tandem, you add an element of specificity that allows you to still do excellent quantitation. And I'd say across mass spectrometry, tandem quadrupoles are among the gold standard instruments for quantitation because of that combination of specificity and sensitivity. Um, Electrospray systems do tend to have kind of measures and countermeasures where you have to evaluate for and compensate for matrix suppression. In both EI, GC-MS-MS, and APGC-MS-MS systems, that's much less of a consideration and not something that's part of the common method development requirements for either of those types of GC-MS-MS systems. The limiting feature being there, maybe the capillary column can't accommodate enough material to become a problem for ionization.
Thanks for that excellent explanation. That's all the questions that we have so far. Thank you so much for this really great presentation today.
Yes, thank you, Doug. Thank you, Jennifer. Thank you, everybody, for joining us today. And with that, we will go ahead and conclude our webinar. And we look forward to everyone joining us on our next ACIL webinar.
Thank you.
Thanks for
Thank you.
joining,
Bye-bye.
everybody.
Bye now.